genome, even of a virus, turns out to be a very long physical molecule. This is the T2 bacteriophage. It's the virus that infects bacteria that we discussed earlier in the Hershey Chase experiment. So this virus has about 164,000 base pairs of DNA in its genome. That's one, I guess two physical molecules, one per strand in the double helix. And the length of DNA is typically 3.4 angstroms per base pair, and so it tells us that the genome of this virus is about 55 microns long. In a virus, you can see right here, there's only about 0.2 microns size of the capsid of that virus. And this problem, this problem of packing DNA is a problem for viruses and also for cells, as an example of E. coli. So the genome of E. coli is 4.6 million base pairs long, and you can see that genome all spread out here from this bacterium right here. So the genome of E. coli is actually a closed circle. But if it were stretched out linearly, it would be 1.7 millimeters long. So it's actually a distance that's big enough to see. Packed into a cell that's just 2 microns long. Another feature of E. coli that you can see up here are these small circles. These are small autonomous plasmids. Many bacteria have these plasmids. They often encode essentially selfish genetic elements, and also can encode things like drug resistance, the way that bacteria, even relatively distantly related bacteria, can pass genetic information back and forth between them. These plasmids are also incredibly useful tools in molecular biology, so we're going to talk about them more. And when you move on to animals, and in particular to vertebrates such as humans, genome size is even larger than it is in E. coli, of course. The human genome has about 6 to 7 times as many genes as E. coli, but it's almost 1,000 times bigger, so 3.1 billion base pairs long. So a couple of things that means. First of all, the linear length of the DNA in a human cell is like a meter long. And of course, in humans, the DNA is broken up into 46 linear chromosomes. So in contrast to bacteria, it doesn't close back on itself. The other thing to note, of course, is that that means that each human gene, if they were sort of spread out evenly across the genome, would get over 100 kilobases of DNA. Now, of course, this is about 100 times the sort of density of genes in the E. coli genome, and it's not that human proteins are 100 times bigger than bacterial proteins. It's actually the case that most of the DNA in the human genome does not encode proteins. And there are two sort of ways that that extra DNA is present in the genome. One of them is the fact that genes in most eukaryotes are interrupted by these intron elements, and so these are non-coding regions with intervening sequences, is what introns comes from. And you can see in these two examples that the white intron sequences that are not part of the final mRNA are actually substantially larger than the blue sequences that are retained in the final mRNA. And in fact, these examples are probably cases where the introns are relatively small. Introns in humans can easily be 10 kb, and in some cases even larger. So what that means is that in the human genome, only a few percent of the DNA is actually coding. Basically, the DNA breaks down to about a third of it is in introns, regions that are inside of genes that are spliced out, and about 2/3 of it is sequence that's outside of genes, and a lot of this is repetitive sequence. That can be either simple repeats, which might be just one or a few nucleotides repeating over and over, or it can be more complex repeats, including transposons and remnants of viruses that are scattered throughout the genome. And so the eukaryotic chromosome I mentioned was a linear molecule. This is a condensed chromosome after it's been replicated, and when it's about to be segregated in meiosis, and there are a couple of sort of landmarks along there. The first is this constriction in the middle, which is the centromere. That's the point on the chromosome where the mitotic spindle is going to attach in order to pull this left and this right-hand strand apart from each other to segregate them during cell division. The other point is that since this molecule is linear, it has two ends. These are the telomeres, and as we're going to discuss when we talk about DNA replication, replicating the very ends of a linear molecule actually causes some substantial problems, and so the telomeres contain sort of special sequences, and the cell actually has a special machinery to deal with these chromosome ends. So another thing you can see just looking at this electron micrograph is that this DNA is not all stretched out in a line. It's actually all tangled up around itself and compacted. And this question of how you compact DNA and the fact that it can get all tangled up around itself is a major limitation on DNA in a large genome, and something that the cell has a pretty elaborate sort of specialized machinery to deal with. And so to start talking about this DNA topology, I'd like to start with sort of the simplest example we can. So on the left is a double strand, sort of double helix, the two strands wrapped around each other, and the way it's drawn here, the idea is these strands are supposed to be anchored at the top and the bottom. So the DNA strands are covalent, or each has a covalent backbone, and so they can't freely untwist and retwist like this. So you can't untwist the double helix and then twist it back up again if it's rigidly anchored at each end. And the same concern arises when the DNA is in a circle, of course. You can't take these two circular molecules and untwist them to make them free like this, and likewise, if you had two strands of DNA, they can't simply wrap all the way around each other without some way to let the two strands cross over each other. And this is referred to as the topology of the DNA, the idea being that we can take either of these two circular molecules and we can distort them in a lot of different ways, change their shape and everything, but we can't actually pass one through the other. And so it's really about the topology of these strands, and in particular, the linking of these strands. So we're going to talk a lot about the linking number of these strands, and here's just showing you on these circles that here, these circles are completely unlinked, so they have a linking number of 0. Whereas here, there's a single link, a single place where this yellow strand crosses around the blue strand, and there's a linking number of 1, and here a linking number of 2 for the two places the yellow strand crosses around the blue strand. So circles always have to have an integer number of links between them. You can have 0 or 1 or 2, and it doesn't make sense to have a half of a link between them. Of course, if you have linear molecules that are partly twisted around each other, you can imagine those things wrapping 1.5 times around each other. But if you had circles, that would leave them sort of pointing off the opposite direction. They in some way have to cross back around each other. Yes, I do. Yeah, so the way that you should count these links, I think the next slide draws this a bit better maybe, is that if you imagine sort of filling in the entire blue circle, so like sort of extending it into a sheet, what you'd see is that this yellow strand goes through that sheet once, and then this point here is, when it comes back around, it doesn't go through the middle of the blue circle, it goes outside. So it goes sort of through the blue circle, outside, through the blue circle again. And so that's 2 links. And so I think what you're pointing out is that, when it comes down to the outside here, that doesn't count as a link because the two are sort of separate. I think that's sort of schematized here in the picture from the book, that if you shade in his blue circle, then you can essentially count the number of times the strand passes inside the circle. 1 here, and then this on the outside doesn't count, and 2 on the inside, and this on the outside doesn't count, and so forth. And this actually has a linking number of 6. So, these strands here are, first of all, one of these strands is just sort of ease of drawing the diagram, has been stretched out completely straight while the other curls around it. In actual DNA, of course, the two strands are wrapped around each other in a double helix. And to remind you of a couple of the most important physical parameters of DNA, first of all, it's a right-handed double helix, and B-form DNA has 10.5 base pairs per turn. And it actually costs a lot of energy to distort DNA away from this 10.5 base pairs per turn. Changing that disrupts the stacking interactions between bases in the adjacent pairs. And so there's a pretty strong energetic cost to doing that. And so what that means, essentially, what we should remember is that the linking number of unstrained B-form DNA is one link, one spiral turn every 10.5 base pairs. And in fact, when DNA is distorted away from this 10.5 base pairs per turn number, it will actually fold back on itself in different ways in order to regain the right sort of helical twist. And this is what's called supercoiling. So I told you how the DNA, essentially the two strands wrapping around each other in a double helix, is one kind of linking. In fact, we call that linking the twist of the individual strands around each other. But another way that a DNA molecule can spiral and link with itself is what's called ride. This is actually the ride of the entire double helix around itself. So that's what's shown here. The twist, so this whole thing is a double helix, and you can see here the whole double helix wraps around itself. This is also called supercoiling. And so the linking number of the DNA is fixed by these covalent bonds. When you have a closed circular DNA, the linking number can't change, but the twist and the ride can change. And in fact, what will happen is the DNA molecule will adopt an amount of ride that it needs to achieve an energetically favorable amount of twist in the B-form DNA. That's sort of drawn here. And then in this example, we have 8 turns of double helix here. And of course, we know that unstrained DNA has 10.5 base pairs per turn, so we can actually figure out that this must be 84 base pairs long. In this example, that DNA has been strained so that it only twists 7 times around itself. So in this example, the linking number would be +8 because there are 8 twists and there's no ride in this molecule. Here, the linking number has been reduced to 7, that there are 7 twists. And in order to return to a favorable B-form conformation, what this DNA will actually do is adopt negative ride. In other words, the double helix will twist around itself, and by doing that, it will introduce more twist into the individual strands and return to a twist of +8. That's the two strands wrapping around each other, and a negative ride of the entire double helix around itself here. So this is an unstrained conformation because the twist has returned to the length divided by 10.5 base pairs, but it's not relaxed because the ride, the whole DNA double helix is wrapped around itself in this twisted figure 8-like shape instead of being a single large open circle. And so in another example, if we have this relaxed DNA, so this is just sort of a single circle laid out in the plane with a twist of +200, that the total linking number of a piece of DNA is given by the amount of twist of those two strands plus the ride of the helix around itself. And if we take that relaxed DNA and we somehow reduce the linking number by 2, the DNA will do is it will adopt this negative supercoiling. So that means that we'll have a negative rise of -2 and a positive twist to get this total linking number of 198. And when the DNA is wrapped around itself like this, that kind of supercoiling, the ride is negative when this is a right-handed helix. So here, if if you look at this, if you put your thumb facing up, and your right hand goes around, the direction that this helix is going up. So you can tell that this is a right-handed helix for negative supercoil here. And to remind you, the DNA double helix is also right-handed when the two strands wrap around each other. That's a right-handed double helix for positive twist. So within this, there's a right-handed helix wrapping all the way around like that. Conversely, if we take this relaxed DNA and add two links, it will positively supercoil around itself. You can see that the helix actually wraps in the other direction, a left-handed way, to give a ride with +2 and reduce the twist to the strands around each other to just 200, so that the DNA is in a favorable B-form conformation again. Yeah? Yeah, so the question is how, in this case, does the linking number change given that? So this could only happen if some process, some enzyme, for instance, in the cell, actually broke the double-stranded backbone of the DNA, passed one strand through the other, and resealed. In fact, we'll talk about in a minute an enzyme that does exactly that. So yeah, so you're right. So changing this linking number requires breaking the backbone of one of those, at least one of those two strands of DNA. Yeah. Oh, yeah. Yeah.
So for a positive supercoiled left-handed helix, is it always positive risks?
So for a positive, positive supercoil means this ride number is positive. Okay. And that is in this whole, like the DNA double helix wrapping around itself, that's a left-handed term. Yeah. And negative supercoil means this ride number is negative. So whenever the linking number is lower than the twist should be to give unstrained DNA, you'll have negative supercoiling and negative ride to make up for that underwinding. And likewise, whenever the DNA has a linking number that's too high for the amount of twist you would like to have in a relaxed conformation, you'll get a positive supercoiling so that the ride takes up some of that linking and the twist matches the amount of twist that DNA would want to be in B form. Yeah. Right, exactly. So the energetically favorable conformation is the twist matches this ideal twist of B form DNA. Yeah. Yeah. Yeah, so ride is when the entire double helix twists around itself like a figure eight. And the twist is a twist of the individual strands one around the other inside that double helix. And both of those contribute to the overall linking number of the DNA. Yeah.
But over here, I'm kind of confused as to how that sort of conformation is achieved, because right now it looks like they're just twisted. So is it just based on the way that they are twisted and changes the way it turns?
Essentially, yeah, I would say in this particular, I would say that drawing out the way that twist and ride can interchange on something that has many hundreds of twists and is in a double helix is pretty hard to do. I think that it's probably it's easiest to remember that when you twist around that when the full double helix twists around itself, that also is a case where essentially it's one more turn of an individual strand passing another individual strand and can make up for some of the difference in twist in the double helix. And then the book just had sort of a diagrammatic picture of some of this, so you can see by electron microscopy just going from this sort of relaxed plasmid circle. I think you saw some of those in the picture of E. coli DNA as it gets more and more wrapped around itself in this highly supercoiled form. this electron micrograph is very two-dimensional, so we can't tell which strand is going over and which strand is going under. So we don't know whether this is positive or negative supercoiling. We can definitely see the double helix crosses over itself many times, indicating this is supercoil. So this I'd like to ask an eye-clicker question, which is basically we're talking about this relaxed circular piece of DNA, and the question is how long is a relaxed circular piece of DNA if its linking number in relaxed form is so you in your minute have to talk amongst yourself about this.
The buzz? Only 19? Okay, blank.
Linking number times base
Is that what it is?
linking number times.
So 200 times.
Isn't it 2,000?
That's not 19.
It would be 2,000.
200 times 10.
All of it. No home. I think it's what you think it's 21. Maybe it's you can't tell though, 'cause you don't know how many Y's this is.
It says relax.
So 8. We go with 8. What is 8?
I think what you do is this. It's this, right? It's like that. Do you think we're proving that we're smart? Wait, put up A, right? What's wrong, DP? Last night.
Yeah.
Put up this. Justin.
Yeah. Divided by 10. No, it's 200 times 10.
This is so cute.
Oh. Okay. Oh.
I'm like so out of it right now. Oh my god.
Yeah, we can just
Don't go on this
No one cares that you're melting down the sprout. It's like not embarrassing.
And the answer is it's 2,100 base pairs. By saying that the DNA is relaxed, that means that it has no rise. And it has no rise when it's able to adopt the linking number, the twist that it would ideally like to have based on its length of B-form DNA. If the twist didn't match the sort of ideal for B-form DNA, then there would be strain, and the DNA would relieve that strain by adopting rise. And so unstrained DNA is 10.5 base pairs for one twist, and there's no rise because it's relaxed. As we just say, there's 200 twists times this 10.5 base pairs gives us a 2,100 base pair piece of DNA. So mentioned that the DNA can, one way to relieve strain from having the wrong amount of twists is to adopt some rise. Actually, another thing that can happen is a small region of strand separation. So where these strands are actually melted apart, there's no twist for this region. If this is about 10 base pairs, then sort of those 10 base pairs are not in B-form, and so they don't count towards the overall length of B-form DNA. So 10 base pairs are melted. Now we have about 7 twists for 74 base pairs, and this is also unstrained. Yeah? Yeah. So the question is, so the ride comes up, the DNA wants to twist around itself once per 10.5 base pairs in order to achieve sort of favorable, I think that one of the dominant forces is favorable stacking of the bases in a way that a lot of high orbitals interact. And so if you twist the DNA a lot, it's energetically costly. It's relatively less energetically costly to just take the whole double helix and twist it around itself. There's some amount of strain and entropic cost that comes up from introducing ride, but it's much less than the energetic cost of having the helix mistwisted. So what will happen is the lowest energy state of the whole molecule will restore the twist to 1 per 10.5 base pairs by essentially tangling the whole molecule up in the way that we saw in those electron micrographs. So the significance of this change in twist when you melt apart the DNA is that actually many of the enzymes that interact with DNA need to melt part of the DNA apart, need to unwind the double helix in part to access those single strands. So one example that we'll talk about later is an RNA polymerase. When RNA polymerase copies the DNA into RNA, it of course needs to melt apart a small region of that double helix in order to have a single-stranded region that it can copy. And so doing that means that it has to untwist the DNA slightly. And actually, in order to make this more favorable to melt apart DNA, the cell actually maintains a small amount of negative superfoil, a small amount of sort of default underwinding in its DNA. And this is expressed as a superhelical density. That the DNA is about 5 to 7% underwound relative to what it should be. And so what that means in practice is the DNA still has that 10.5 base pair twist per turn, but it's all twisted around itself and negatively supercoiled by default. And when, let's say, RNA polymerase goes to melt apart a small region of DNA, the cell will lose some of that negative supercoiling ride to allow RNA polymerase to melt apart that small region of the genome. Yeah? That's an excellent question. So eukaryotic DNA is not circular, and in fact, sort of related concern comes up in the E. coli genome, which is a huge circle, and actually what happens, and I think this is kind of drawn on the next slide, is that there are what are called topological domains, where a protein structure will anchor big pieces of DNA. So it's sort of more like the example I showed where they're essentially just anchors on each side of the double-stranded region. There'll be a big loop in E. coli. It's about, I think it's about 10 kilobases for an E. coli domain, or domains of eukaryotes are much larger. But essentially by having these protein complexes that are anchoring the DNA, and again, those are those big loops that you see coming out of the chromosome, those are regions where topological strain can't move outside of them. So in a simple circle like a plasmid, you don't have those sorts of anchors. Actually, so this is relevant to this idea of transcription. In that so I mentioned that, of course, the polymerase melts apart some of this DNA when it binds, and it also sort of moves along, which means it has to untwist ahead of it and twist behind it, and you actually build up overwinding ahead and underwinding behind. One of the reasons a cell maintains sort of a default amount of negative supercoiling is so that this overwinding isn't actually overwinding, it's more like removing underwinding, and that sort of provides some amount of, it's less energetically costly to do that. And that happens both in E. coli and the E. coli genome, where it's anchored in these regions, as well as in the eukaryotic genome. So, of course, a lot of this, the question is, how does the cell eventually deal with this changing the winding, especially in these cases where the DNA starts to become overwound and underwound. And one thing to remember is that breaking even one of the two strands in the backbone, which is called nicking the DNA, means that it can remain double-stranded and becomes topologically unlinked. So now we have this little break here. The two strands can freely move around each other, and so you can flip back and forth between these two conformations. There's no longer a covalent bond in the way. And so unlinked nicked DNA has no linking number defined, because these can twist around each other, 1, 1.5, 2, and so forth times. And it can also freely adopt its lowest energy state, where it's relaxed, has no ride, and it takes on its preferred twist of a 1 turn per 10.5 base pairs. And in fact, when we saw this electron micrographs and all this DNA isolated, probably the reasons that it was all spooled out in these sort of smooth loops instead of all wrapped around itself is that it had gotten nicked in the process of isolating that DNA, and so all of the supercoiling was released. so that the cell has enzymes that do exactly this, that they nick the DNA backbone, allow the two strands to pass between each other, and then reseal the break. These are called topoisomerases because they take the DNA molecule in one state, one topological state, break a bond, reform the exact same bond. So they form molecules that have the same structure, except for this topological linking between the strands. So they change the DNA between two different topological isomers. And so type 1 topoisomerases transiently nick the DNA and they break one strand, pass it through itself, and then reseal it. And so by doing that, they change the linking number up or down by 1 unit. And in fact, the mechanism of these type 1 topoisomerases is simply this nucleophilic attack mechanism that I told you was the only chemistry we really need to learn about the DNA backbone. So there's a tyrosine residue. The oxygen on the tyrosine residue forms a nucleophilic attack on the DNA backbone of one of the strands and actually remains bound to that DNA that it does not let go. It doesn't want to leave this broken DNA behind. It holds on to it while one strand passes through the other. And then it simply reverses the reaction to reseal the backbone. It's preceded by the nucleophilic attack of the 3' OH back on this tyrosine phosphoester. And so by doing this, you can interconvert DNA. And because there's no energy involved in this process, it's always relaxing the DNA towards its lowest energy form. In fact, we can see that on what I think is a pretty neat demonstration of a few different things. First of all, the action of the topoisomerase is also the fact that there really are integer linking numbers. And so what's happening here is this highly supercoiled form of DNA moves very easily through a gel, whereas this floppy, relaxed form migrates much more slowly. So the relaxed DNA is up here, the supercoiled is down here. Told you that cells typically underwind their DNA, and so when you isolate unnicked DNA from cells, it's supercoiled and migrates like this, but even as a result simply of handling it, some of it ends up nicked and relaxed. When you take this DNA, most of which is supercoiled, you add a type 1 topoisomerase, it allows it to freely interconvert between different isomers, and the result is to drive it towards this relaxed form, and you can see all of the intermediates with linking number 0, 1, sorry, a ride of 0, 1, 2, and so forth down here. So I told you that there are these type 1 topoisomerases. Sort of suggests there's also a type 2 topoisomerase. And a type 2 topoisomerase is a pretty easy mnemonic. That a type 2 topoisomerase breaks both strands, breaks 2 strands of a double helix, takes another entire double helix and passes it through. So it moves one unbroken double helix through another entirely broken double helix. So a type 1 topoisomerase breaks 1 strand, a type 2 topoisomerase breaks 2. Because it's breaking 2 strands and moving 2 other strands, if you treat a supercoiled single piece of DNA with a type 2 topoisomerase, it actually changes the linking number by 2 because it's moving 2 strands. Another feature about a type 2 topoisomerase is that it can untangle separate DNA molecules. If you somehow ended up with 2 circles, one wrapped through the other, a type 1 topoisomerase can never resolve this because it only breaks a single strand of the double helix and moves the other strand of the same double helix through it. So the cell couldn't fix this with a type 1 topoisomerase. Unfortunately, it has these type 2 topoisomerases that take an entire second strand of DNA, move them through, and let it go on the other side. This is called decatenation. When you have 2 circles, one through the other, it's called a catenate. And another point is that this enzyme activity is ATP dependent, and in bacteria there's a type 2 topoisomerase called gyrase that uses the energy of ATP to add negative supercoiling to DNA. And so this activity of gyrase is what's responsible for the fact that bacterial DNA by default is slightly underwound. Gyrase uses the energy of ATP to underwind it, and then when you go in and need to, for instance, transcribe that DNA, some of that energy is essentially stored, that ATP energy is stored in the twisted DNA and is in the writing DNA and is released to help RNA polymerase melt apart the two strands. In addition to these effects of topoisomerases, there are other ways. Yeah. So this whole process is a cycle where a type 2 topoisomerase changes the linking number of the DNA. So gyrase is one example of a type 2 topoisomerase. So this type 2 topoisomerase is a eukaryotic type 2 topoisomerase. Gyrase has the same mechanism. It uses ATP to change, to run through this whole cycle. So the whole cycle is the thing that ultimately changes the linking number, isn't it? You break two strands, pass two other strands through them, reseal those two strands. And so that's why it changes the linking number by 2 and not 1, because you're moving two strands. And it also allows you to untangle DNA that either closed circles or giant loops that have gotten twisted through each other. You can separate them with a type 2 topoisomerase. So another way that the topology of DNA can end up changing is the presence of what are called intercalating agents. So if you, in a molecular biology lab, have ever used a dye Ophidium, or any of the sort of related dyes used to stain DNA, it turns out that these things actually slip in between the base pairs of DNA. And actually one result of this is that they decrease the amount of twist that the DNA wants to have. In fact, if you totally saturate a molecule of DNA, you can dramatically reduce the twist. So it's hard to add that much Ophidium. It does other things. When they slip between the bases, the dye becomes fluorescent. That's why we use it to look at DNA. But it's also a useful experimental tool to study topology because you know that it decreases the twist. And in fact, you could use this to look at, for instance, the action of topoisomerases in sort of example where if you had relaxed DNA, here is the same example we've been using, a 2100 base pair piece of DNA with a twist of 200 and no rise. It's relaxed. If it's a closed circle and you add Ophidium to it, what you'll actually see is that it starts to positively supercoil. The DNA wants to have less twist in its relaxed conformation, so it's no longer B-formed DNA. And so if you have some sort of specific reason to know that DNA does not want to be in B form, for instance, we've added Ophidium to it to decrease the ideal twist. So the first thing that will happen is it will adopt this positive supercoiling as the DNA twist changes and the rise changes to match to preserve the linking number. But now if we add a type 1 topoisomerase, we can actually relieve this rise so the DNA can become relaxed again, but at this lower twist. And then if we remove both of these, it would actually snap back into a negatively supercoiled state. So we've now returning it, so we've removed the Ophidium, returned it to this, this should say negative supercoiled, returned it to this negatively supercoiled state as it underlines. DNA in bacteria, I mentioned, is negatively supercoiled. And this kind of negative supercoiling where it twists around itself like this, and you can see that this is right-handed twist of the double helix around itself. It's called plectonemic supercoiling. And E. coli is introduced by gyrase, the type 2 topoisomerase that uses the energy of ATP to change the DNA like this. And in addition to plectonemic supercoiling, when the DNA wraps around itself, there's also what's called solenoidal supercoiling. And this is when the DNA is wrapped around some other sort of spindle. You can see the interconversion between this plectonemic supercoiling as you start to take the strand, unwind it from around itself, and instead wrap it around something else here, till eventually have this solenoid. So you can freely interchange between these two different topologies. And in bacteria, this negative supercoiling is all this plectonemic type, the backbone wrapping around itself. In eukaryotes, though, the supercoiling is actually of this solenoidal type because the DNA in eukaryotes is actually wrapped around histones in the chromatin. So eukaryotic DNA is packaged into these histones, packaged into this chromatin, where you can see that the double-stranded helix wraps in a solenoidal way around these histone proteins. In fact, the definition of a chromatin is a string of nucleosomes, and those nucleosomes are the DNA wrapped around these histone proteins. And so eukaryotes actually maintain negative supercoiling without the activity of a gyrase-like enzyme, simply by this solenoidal wrapping of the DNA around the histone. In fact, this is sort of an example. If you take naked DNA and you add a histone to it, you'll get the histone binds DNA very well, and so the DNA will solenoidally wrap itself around the histone and then make up for that negative supercoil with plectonemic ride in the other direction. And if you then take this and introduce it to a topoisomerase, you'll relax out this positive plectonemic supercoiling and have a circle that is relaxed except for this one negative supercoil wrapping around the histone protein. And so histones are, I'd say, really fundamental to thinking about all the things that chromatin, the DNA, does in eukaryotes. First of all, just the effect of packaging the DNA, and also the negative supercoiling that they introduce. And so the nucleosome itself is about 146 base pairs of DNA wrapped around the nucleosome. It's a very uniform distance, the amount of DNA that's really tightly bound to the histone itself. There's also a much more variable bit of linker DNA between those, and so the distance between nucleosomes is typically a total of about 200 base pairs. You have about 50 base pairs of linker DNA between each of these individual histones. Yeah. Histones are only for eukaryotes, definitely. And so plectonemic conformation can arise in eukaryotic DNA. There's no reason that it can't, but if you sort of look at the genome at steady state, the bacterial genome has a substantial amount of plectonemic negative supercoiling. The eukaryotic genome has very little plectonemic negative supercoiling, but quite a lot of solenoidal negative supercoiling from the DNA being wrapped around the histones in the chromatin. Yeah. Yeah. So it's, again, I think you essentially just, the easiest way to see this is you just had some physical thing that you could sort of wrap in a plectonemic way and then start to pull it apart. It just turns out that a right-handed plectonemic ride, if you start to pull that apart and wrap it around something else, that you need to wrap it in a left-handed way to get the same thing to happen in solenoidal ride. So it's, I mean, I would say that you could try to get something like, if you have like something like a telephone cord, it's a classic thing to do this with, or maybe something like a rubber band, you can wrap it up around itself, and then you can take it, unwrap it, and wrap it around something else, and you can see what direction you need to twist it in one to the other. But I think that probably is something just to remember, that that negative supercoiling is a right-handed plectonemic helix or a left-handed solenoidal wrapping. Yeah. Pardon? So a gyrase is a type 2 topoisomerase in bacteria that uses the energy of ATP to introduce negative supercoiling into the bacterial genome. And eukaryotes do not have the gyrase. So they do have type 2 topoisomerases. They don't have one that does that function. So eukaryotic DNA in chromatin, of course, the DNA, and then also these histone proteins, which are very abundant and very highly conserved proteins. Each histone has an octamer of individual polypeptides, so 2 each of what are called the core histones, H2A, H2B, H3, and H4, with 146 base pairs of DNA in 1.67 negative supercoiling turns around it. Here's sort of a schematic picture of what it looks like. Here's actually a space-filling model with the proteins in the middle, DNA wrapping around them. Again, a picture here, a top-down view. So histones bind DNA well. DNA, of course, is acidic, which means that it's negative at neutral pH. And in order to bind DNA well, histones are very basic proteins. They're positive at neutral pH. So they're rich in arginine and lysine residues, and that promotes sort of favorable interactions of the DNA with the histone. Histones, in addition to this very stable core structure drawn here that the DNA wraps around, also have unstructured tails that are drawn here. These are present at the N-terminus, the beginning of each of these histones, and they're sites of post-translational modification in ways that control the expression of the DNA that's wrapped around that histone. There are a huge number of these modifications that have been identified. I would say the ones that come up the most are serine phosphorylation, lysine and arginine methylation, and also lysine acetylation that happen all along these tails sticking out. So there are enzymes that come along and modify these tails, and other proteins that come along and bind these tails based on how they've been modified, and that in ways that we'll talk about later can affect whether or not that gene is transcribed and how that DNA chromatin might be made more accessible or less accessible. So beyond the sort of the beads on the string organization of the genome into nucleosomes, there is a certain amount of higher order organization for eukaryotic DNA. It's less stereotyped, but often these nucleosomes assemble into what's called a 30 nanometer fiber. Where each of these yellow spheres is one of these nucleosomes wrapping around like this. And then this 30 nanometer fiber can itself be strung into these domains. So here, there's essentially a protein scaffolding of some sort. Each of these big loops, these are the topological domains of the eukaryotic genome. And then, especially in mitosis, even these loops can be gathered together into this form we see here, keeping in mind that this whole thing has been condensed from something that's about a meter long to something that's about a micron long. So there's a huge amount of compaction that goes on. The first stage is this wrapping the nucleosome, then this formation into this these fibers and these domains, and then this last step, this condensation, is a result of what are called SMC proteins. So the sort of the configuration of SMC proteins in chromatin is much less clear than the organization of nucleosomes, but it's pretty clear by electron microscopy is that they have this form where they have a head and a hinge. The head has ATPase activity, and that ATPase activity controls the opening and the closing of this SMC ring. And they also have these very long arms that are coiled rods. Coiled rods close DNA inside of it. And there are 2 different models, and people don't actually know for certain how many DNA molecules are closed by these actual cells, and exactly how this participates. But these are the molecules that are important to the highest order compaction and condensation.